advancements that we made on the algorithm side in the space of graph neural networks, and then also thoughtful use of those methods and application to problems in the interface of molecular data and therapeutics. So let's start. Science crucially depends on existence of scientific instruments, and in the past those instruments were primarily physical devices such as microscopes that facilitated discoveries. And now we are in the era of modern data-intensive sciences, as we have just discussed before the beginning of this talk. And so in many ways, we need new kind of tools, new kind of instruments that many in the field and we will depend in vital ways on knowledge discovery from data. So this is a great vision that is shared by many in this field and probably many on the call, but why is it so challenging to realize this vision? And there are a number of reasons that prevent us from realizing that vision in the context of biomedical data. And I will be mentioning here two major challenges that I'm thinking about at the high level. So first is that for most of biomedical problems, it really is not the case that there is a single data set that can explain the entire complexity of the phenomenon that we are studying. So we need to integrate heterogeneous confounded data that span from molecules all the way to societies level data. And so that such representations nowadays don't fit naturally into one large flat table or data set that is predominant view of how machine learning thinks about how we think about the data set in machine learning. So data sets are multi-scale, go from molecules to populations, heterogeneous in the sense that they they might come from experimental readouts. Some are curated or self-reported, and because of that, they're also confounded in ways that we need to consider, because they come from different technologies, different measurement platforms, or different clinical sites. Second important challenge is that in biomedical settings, it's not only about eureka and we solve the problem and here is our best and possible AUC score or measure of accuracy, but it's really important to be able to have to be able to generate actionable predictions or actionable hypotheses that can directly inform downstream experiments. And so I'm taking this figure on the right from one of the Jianxiu and Tri Ideker's paper about how to now transform these black box ML models into white box models whose predictions we can interpret meaningfully. In much of my research, we are using networks or graphs as a general language that can bring together heterogeneous data sets. So the way I think of network is that they allow us to represent information in interactions at the level of populations or the level of groups of individuals and various kinds of interactions that might exist between them, all the way down to interactions between various phenotypes in disease or diseases within an individual, and then interactions between molecules or cells at the cellular level. So we can connect these different scales to each other, and that gives rise to this multi-scale network representation that can bring together rich multimodal data. So networks for me are a mechanism that allow us to computationally operationalize these large data sets coming from a variety of resources. And the natural question to ask then is, once we have these networks or graphs on the left, how can we design powerful methods for learning that can generate actionable predictions? And so those would be predictions that predict properties of nodes that appear in the input graph, such as various functional properties of proteins, or if patient outcomes if those are represented in our graph. Discovering new associations or relationships between entities that appear in the graph, predicting properties of entire molecule graphs, or being able to understand the landscape and similarities and differences between these entities. And so graph neural networks, or more broadly graph representation learning, has emerged as a prevailing paradigm in the last 4 years for learning and reasoning over graphs. And And in one slide, I will try to summarize or the key technical principle that is behind graph neural networks. And that core idea is the notion of network embeddings or graph embeddings. So the idea is that if we have our graph on the left, which is the graph that represents our particular problem that we are studying, the graph neural networks specify deep transformation functions, which is here denoted as function f, that maps nodes from the input graph on the left to some low dimensional embedding space. And how that embedding or that mapping to the d-dimensional space is done, it is done such that if two nodes u and v in the input graph are considered to be similar, meaning they have similar interaction patterns, they have similar network neighborhoods in the combinatorial structure of the graph, then they be embedded close together in the embedding space. So the goal of this graph neural network functions f is to learn and optimize an embedding space that can faithfully represent the combinatorial structure of the graph. And so a variety and really a large number of variants of graph neural networks were developed and proposed in the last 4 years that operationalize these different notions of what it means that two nodes have similar network neighborhoods in the graph and how they can be embedded in the embedding space. But the core notion is that we learn these vector representations of nodes, of edges, of even larger graph structures. And once we have those vector representations, then we can use the power of linear algebra and rely on the large body of existing work in ML and linear algebra to utilize those embeddings in interesting way. Okay, so graph neural networks, to put them into perspective, are complementary to two other classes of very powerful neural networks, and those are various variants of convolutional neural networks for images and recurrent neural networks and LSTM variants for sequences and for language sequential data. So what graph neural networks allow us, that we can now apply deep learning much more broadly, not only to image data, not only to sequence data, but to any graph or network in biomedical domain. And so this creates lots of new opportunities in biology and medicine. So my plan for today is the following. I will highlight two recent methods that we have developed this year for embedding subgraphs and for transfer learning on graphs. And I will describe how those two methods have enabled us to tackle interesting problems in the space of therapeutics, to identify safe, effective drugs, and to tackle emerging diseases and diseases we have never seen before, and how we can really make some useful predictions for those. And finally, I will conclude with a new initiative that we have recently started that is called Therapeutics Data Commons, and I will just mention it and provide some pointers. Okay, so let's start with the first topic. So in the first part of the talk, I will outline the methods subgraph neural network, which we just published at NeurIPS this year, and then I will describe how we use the method in the context of identifying effective and safe drug treatments. So the primary motivation for this part of work is essentially is a problem of drug development and discovery, which is very familiar to people on the call here. And so as you know, this process is extremely long and expensive. And in fact, less than 7% of drugs that for example, oncology drugs in clinical 1 trial phase are then eventually approved. So there is a huge failure rate of drugs that need to go through this particular funnel of the drug development phase, and only a very small number of compounds eventually get approved, and it takes over between 12 and 16 years on average to design a new drug completely from scratch. And so there are many alternative strategies that researchers have been exploring, including the ideas of how it can be possible to extend the use of drugs that we already have or combinations of those drugs for new users much more quickly and in a rapid way, and also more efficiently, and such that the process is faster and cheaper. In the ML's language, the core question that needs to be solved there is trying to understand what molecule or drug will have has a promising positive therapeutic effect on the disease. At the very high level, the way we can think of this problem is that we have a large bipartite graph. We have a set of drugs on the left, in the left part, and then we have a large number of diseases on the right. And so these black edges between drug and diseases here show known indication information. The question then becomes, how can we leverage that information effectively to discover new indication relationships between drugs that might be drugs that are already on the market, or those that are being developed for diseases, and predict what diseases an animal might treat. The key insight that we will be leveraging here is this notion of subgraphs. So what that means is that we want to move away from representing drugs and diseases as kind of simple nodes in the bipartite graph, as I shown in the previous slide. Instead, we want to construct large the biological graphs, and here is shown a pictorial example of it with these white nodes and black edges, and then think of drugs and diseases as subgraphs that are overlaid on that graph, or on that underlying graph. So we will think of diseases as subgraphs of rich protein networks that are defined on disease-associated proteins, and drugs as subgraphs of their drug targets as well as off targets, to the extent that that information is known. So that now means that our setup is that we have this underlying proteome scale interaction network on that network we overlay a large number of disease and drug subgraphs. Now in the pharmacological space, it is known, and we often can read that a drug is likely to treat a disease if it is close to the disease in pharmacological space. So that sounds great, but what does it mean being close to the disease in pharmacological space from computational standpoint? How can we computationally operationalize that? So our idea will be then to use the power of embeddings to operationalize this concept of closeness. And we will learn embeddings for every drug, for every disease, and then we will see, well, what drug embeddings are close to what disease embeddings, and that will inform our predictions. So a natural question to ask now, if you follow the previous slide, is why subgraphs? And so there are many reasons for why we think subgraphs are the better way of modeling this complex biological phenomena than simply representing them as flat one-dimensional nodes. And one of the reasons is the following. So recently we've been involved in the COVID task force at Harvard and our partners at federal agencies, and we used our methods to identify promising drugs that would inhibit SARS-CoV-2 virus. And what we found is that 76 out of 77 methods, drugs that were predicted by our methods and successfully validated in the wet lab, do not directly bind to proteins targeted by the virus. Instead, those drugs rely on network-based actions that could not be identified by simply looking at alternative non-network methods. And we call those drugs network drugs, because in order to identify and model them, one really needs to think about the entire structure of drug targets, how they are related to each other. And so it's not sufficient to simply represent drug as an individual nodes. subgraphs are a better way of representing that, as also demonstrated by this, which I will discuss more later in the talk. Okay, so that is a motivation for why we want to use subgraphs and learn subgraph embedding. So how exactly now we do that? And so our goal is that we want to have a method that will learn subgraph embeddings such that the likelihood of preserving subgraph topology is maximized in the embedding space. So what that means, if I have two subs as I and as J that have similar topology, they should get embeddings that are also similar. Those two subgraphs should be embedded close together as points in the embedding space. And SubGNN or subgraph neural networks are a new representation learning framework that can achieve exactly this goal, that can learn and generate embeddings for subgraphs, such as shown here in this example at the bottom of the slide, where we have the graph which is sprinkled with 5 subgraphs, S1, S2, S3, S4, and S5, and then SubGNN can generate an embedding for each of those subgraphs by taking into account a variety of important aspects of subgraph topology. In particular, those are asking how does the network neighborhood around each subgraph look like? Or what is the structure and network motifs and shapes that are found within each of the subgraphs? So here we can see we have a triangle and another triangle and a square. And then position, where is the subgraph located within the entire space of the graph? We would say S1 is on the periphery of the graph, but S4 is closer to the core of our underlying graph, right? Because it's here in the center of the full graph, which is just shown here pictorially. Okay, so the overview of SubGNN is that it has two key parts. And so part one is the part where we specify a transformation function that is called neural message passing function that can hierarchically propagate information along edges in the underlying graph. And it does so by specifying some form of neural messages that get propagated from the rest of the graph into subgraphs that we want to embed. And those messages are aggregated to generate embeddings. And in part two, SubGNN specifies how to do that propagation effectively to capture different types of topology, position, neighborhood, and structure, as I described in the previous slide. So I have two slides of a bit of more technical details. And then I will return to high-level description. So to unpack part one a bit more for those who are not familiar with graph neural networks, what we do is the following. So we specify neural messages, and we specify a separate type of neural messages for each of 3 aspects, properties of subgraph topology: position, neighborhood, and structure. And then what we do is within the underlying graph, we sample a number of anchor patches. Anchor patches are helper subgraphs that we don't care about in the sense that we would want to embed them, but they help us learn better embeddings for subgraphs of interest. By sampling anchor patches and then asking how do these anchor patches relate, for example, to this subgraph in red, we can answer questions whether the S1 subgraph is closer to periphery of the graph, is it close to the center of the graph, what does its local neighborhood look like, and so on. And so in this part one is really powered by this neural message passing scheme that has different types of that specifies a propagation algorithm for sending messages which are vectors between and exchanging those vectors between anchor patches and subgraphs. And that exchange is done by defining anchor patches denoted by A, then measuring similarity between anchor patches and our subgraph of interest, and then every subgraph receives a number of messages from a number of anchor patches. So we aggregate that information and then transform it, pass it through a nonlinear activation function to get a subgraph embedding. To get property-specific representation or embedding of a subgraph or its component. In the second part, we repeat the process that I just outlined, but we repeat it in 3 different channels, each channel designed specifically to capture distinct subgraph property, position, neighborhood, and structure. And so all together, the way to think about this is that we have the underlying graph, such as protein interaction network, that is overlaid with many drugs, with many disease subgraphs. Each of drug and disease is represented as a subgraph. SubGNN, what it will do, it will learn what is the best way to aggregate information, to pass information from subgraphs to this underlying protein interaction network, pass it along edges in the network, and aggregate messages back at the subgraph level. And so this idea also mimics the notion of how the effects of drugs are not limited only to proteins to which the drug directly binds, but are really propagated along edges of the underlying graph. This is exactly what the model is also operationalizing mathematically. Okay, so that is the overview of the method. Let's now look at what kind of questions we can ask and answer with this method. So the first analysis, or the first experiment I will describe, is related to drug repurposing. So here we worked with our collaborators to pull together a large protein interaction network from 15 biorepositories, information on drugs and diseases to construct drug and disease subgraphs, and also information from our collaborators at the FDA and Drug Central on medical indications and contraindications to construct large representation that looked like that. So we had the underlying protein interaction network. Onto that network, we have large number of diseases, disease subgraphs, and large number of drug subgraphs. Exactly the kind of structure that I showed in the beginning of the talk to motivate this part. So once we had this data set, we used SubGNN to learn embeddings for disease and drug subgraphs. So our input data looked like thousands of disease subgraphs, thousands of drug subgraphs connected based on known use of drugs for diseases and also contraindication of drugs for diseases. And SubGNN learned embeddings for drugs and diseases, and then being able to predict whether a particular drug C should be indicated or contraindicated for a particular disease, which corresponds to predicting these edges here between drug disease subgraphs. So we did lots of classic types of analysis we do in machine learning, including ablation and cross-validation studies. I will not go into details there. I will highlight one particular experiment that we did with Spark Translational Research Program at Stanford School of Medicine. And that program has repurposed a large number of drugs in the last 20 years. And essentially taking existing drugs from the market and repurpose them, expand their use for novel diseases. And that center gave us a list of long list of drug disease pairs where a particular drug was repurposed, expanded, indicated for a disease. So if we take an example, fourth row from the bottom, it says carvedilol used for Chagas disease. And so that for us was ground truth information that we used to evaluate the model. And we asked the model, great, if the model has not seen this data before, how highly would the model rank carvedilol as in the list of all drugs that we have for Chagas disease? And the model ranked carvedilol as 9th most promising drug for Chagas disease. And you can see in many other instances, the ranks were the ranks are relatively low, meaning that the drug that was successfully repurposed within the center was actually predicted to be very close to the top of our priority list of drugs, which is very good. So that has led that has encouraged us to do more exciting new follow-up research that we have on prostate cancer and schizophrenia. We're looking at designing combinatorial treatments of prostates with that also minimize incontinence as unwanted side effect. And schizophrenia, where we really have multi-scale networks that have also include data on what is happening below the neck, not only above the neck, which was what is the type of data that was so far mostly considered for the problem of schizophrenia. Okay, so in identifying effective drug treatments or possible indications or contraindications, which is what I just talked about, an important question, an important second endpoint is that of safety or unwanted adverse events that are associated with the drug. So here I will mention one example of a study that was enabled by the architectures and neural network that I described before. And in particular, we were looking at combinations of drugs. So combinatorial treatments or polytherapy refers to the setting where patients take multiple drugs to treat complex diseases or coexisting diseases. This is a very common situation. 40–60% of people of over 65 years take more than 5 drugs. It's not uncommon to encounter patients that take more than 20 drugs, and people that take many drugs at the same time, they are at much higher risk of developing unwanted adverse events. And that is extremely hard or impossible to study in a large systematic manner through clinical trials, because simply there is an explosion of possible combinations of drugs. Not all of them, it's simply not feasible to test all possible combinations before those drugs are being prescribed together in patients. And because of the challenge, the increased risk of developing adverse events when a patient is on taking a combination of drugs, this presents many challenges, both from individual patients as well as for healthcare system that now needs to treat those adverse events, and that is an important problem. So when I said that patients are at high risk of developing adverse events because of unexpected interactions between drugs that they are taking, what I meant is the following. Imagine we have a system where with 2 drugs, we have blue and red pill. When a patient takes blue pill, the desired expected therapeutic effect is achieved, no substantial side effects. When a patient takes red pill alone, again, everything's okay, no substantial side effects. However, when a patient takes blue and red and blue pill together, the patient experiences side effects. And so why is this indicates unexpected interaction between drug? Because if the effects of blue and red pill would be completely independent, then we would not expect to see these adverse events in patient populations that take red and blue pill together. So the question will be, how can we use our graph neural networks to predict how likely a particular combination of drugs lead to a particular side effect? And so to answer that, we have put together a lot of data from FDA and their national adverse event reporting system, and in particular that was data set from 10 million reports that FDA has on adverse events. And we constructed a large polypharmacy network, which is a network where we have 2 types of nodes. We have drugs represented by green triangles. Different types of interactions between a pair of drugs are represented by edges between drugs. So the fact that there is an edge of type R1 between these 2 drugs indicate that when patients take the 2 drugs that are denoted here by green triangles together, they will experience with some probability a particular side effect R1. and that probability is weight of that edge. And in addition to drugs, there is information about how those drugs target proteins and how those effects propagate within the protein interaction network. So we constructed this large graph, and then we used and trained our graph neural network on that polypharmacy networking graph with the goal of asking questions that are posed by domain experts. For example, domain expert can ask how likely will a form of statin and ciprofloxacin, when taken together, break down muscle tissue. When a user asks this question, behind the scenes, the graph neural network is asked to predict the probability of an edge of type breakdown muscle tissue existing between the two nodes in the graph that represent the drugs in question. And so we trained the model on this large polypharmacy network, and then we again did a number of experimental, empirical, computational validations and cross the validation tests, and also looked at new predictions. So this is what I'm showing on this slide, where we trained the deep model on data generated prior to 2012, and then asked what are predictions that the model is most confident about, and whether any of those predictions have been confirmed after 2012. So this is known as a temporal split of the data into training, validation, and test set. So what is shown in this table here are 10 most promising or 10 predictions that the model was most confident about. The way to read those predictions is that each prediction is a pair of drugs and the type of side effect that is predicted to be associated with that pair of drugs and cannot be attributed to either drug alone in the model. And so we found lots of evidence in published literature published after 2012 for top predictions made by the model. There are, of course, some novel hypotheses that the model has generated. And so since then, we have engaged and pursued avenues to clinically validate predictions made by the model in health electronic record data, as well as deploying the model in a few clinics where physicians now have side by side the decagon model for polypharmacy side effect prediction and existing drug-drug interaction markers on their computer when they are actually discussing discussing a situation with patients, so during their patient visits. So far we have several promising predictions that we were able to successfully validate using using surrogate measures for drug interactions, including lab values and interaction markers. Okay, so this concludes this first part of the talk, and in the second part of the talk, I will move away a bit from drugs and focus slightly more on diseases and the notion of, in particularly, tackling some of the hardest cases, which I think are also very exciting for machine learning in the context of biology and medicine, which is really how can machine learning inform experiments and downstream validation of and kind of downstream research work for most challenging cases where we think about phenomena or which might be drugs, diseases, patients that we have never seen before. So when the models are tasked to making predictions for very new objects, for patients that are hard to diagnose and cannot and potentially have rare diseases, or with diseases that we didn't know much about just a year ago. And so the primary methodological concept here is that of meta-learning or transfer learning, which I will outline first, and then I will talk about applications of the model in the context of never before seen diseases. Okay, so you have seen this slide before, but this is a slightly different version of it, and this subtle difference is actually very important. So again, imagine we have the we have a bipartite structure of our data set. We have a large number of drugs and molecules on the left and diseases on the right. So now one of the diseases on the right is a very new disease, disease that we have not yet, we don't have any good treatments for. We don't know much about it. So our goal will still be predict what drugs might treat that new disease. But what is really challenging here is that there is no existing black edge that would connect this particular disease to drugs. So really, there are no good treatments for this disease. This disease is very different from what we see from many other diseases that are better understood. So how can we really make these initial predictions for emerging diseases and pathogens? And the motivation for this work that I will describe in this part of the talk was certainly the ongoing pandemic that we are dealing with, where we observed that traditional iterative development, experimental and clinical testing and approval of new drugs is simply not feasible. And so we resorted to drug repurposing to identify drugs that have likely therapeutic effect in COVID-19 patients. More concretely, we have formed together in Harvard ecosystem a task force that was established on March 23rd of this year, when we started with the collection of the data and then deploying our AI toolbox to these data sets to identify a promising drugs for COVID-19, and I will explain how that can be done even though there were no drugs or successful treatments known for the disease before. And by April 19, we shared finalized predictions by the model, shared those predictions with research community, and then engaged in experimental screening of predictions on monkey and human cells. And so by June 15, we have validated predictions and identify some new drugs. So I will explain how we were able, and how machine learning and graph neural networks in particular enabled this fast deployment of AI algorithms, and really to accelerate and expedite this process and compress work that would be normally done in months, just doing it in weeks or even days in some cases. Okay, so let's now return for a moment to ML context. So the main challenge that needs to be solved is that we need to use and have machine learning algorithms that can generalize to new phenomena, that can generalize to new diseases that are not in the training set at all. And so this generalization to new phenomena, also known as auto-distribution generalization, is typically very hard. Why? Because prevailing methods require abundant label information. However, labeled examples are very often scarce. And I'm using COVID-19 as an example here, but this particular problem is pervasive in many exciting areas at the frontier of biomedical research. When we are, for example, when we are tasked with modeling novel drugs in development, when one wants to model rare diseases or identify and diagnose patients that are hard to diagnose because they have particular, again, rare disorders or disorders that are not classified in medical settings. So prevailing methods, what assume is that we have these graphs and networks, but they're already densely labeled. So labels here are denoted as colors on the nodes. What really happens in the world is that we have really these large graphs, but they're not so densely labeled. We just have many cases, a handful of labeled examples, a handful of successful treatments, if any at all, that can be used to supervise and train the models. So how can we tackle this challenge? And so when labeled data are scarce, algorithms really need to be successful at learning from prior experiences and need to be able to form inductive biases fairly quickly to quickly adapt to new machine learning tasks. And so the general field of or area of research that studies these problems is transfer and learning, or meta learning. And so I will describe a particular algorithm that was also published at NeurIPS this year called GMeta, which is meta-learning algorithm for graphs that allow us to advance algorithms to train more with less data and very quickly learn about never before seen phenomena. So how can we design these good meta-learners is the question that I will try to answer in the next few slides. So again, a bit more technical details for those in the audience who are interested in graph machine learning. So the key challenge of today's graph machine learning, graph neural algorithms is that they rely on this propagation of neural messages along edges of the graph, which is what I described before in the first part of the talk. That works fantastically well when we have lots of label data that we can diffuse or propagate along edges of the graph. But when we have very little data and then we have very large number of graphs, that propagation is highly uncertain. It's unreliable. So instead of passing those messages, neural messages, and diffusing labeled information throughout the entire graph, our key idea of GMeta was that what we will do instead, we will extract local subgraphs around target nodes and enclose labels. And then we will apply graph neural networks individually to each of those local subgraphs. So again, you might have seen that there is this recurring topic of subgraphs. So what is the value of subgraphs here in contrast to what was the value of subgraphs in the first part of the talk? So in the first part of the talk, the goal was to embed subgraphs. Here, we are not embedding subgraphs at all. Our goal is to leverage subgraphs to improve our ability to make good predictions. And the way to motivate this work is to think about, okay, so what allows graph neural networks to really make good predictions on graphs? And we typically think that there are two sources of predictive power for machine learning applied to biomedical networks. The first is label propagation. We assume that nodes that have the same label, the same property, are nearby in the graph. That has been shown over and over and over again, by, for example, indicating that proteins in networks that directly interact or that are relative proximal in the graph, they are more likely to also share certain molecular functions. And so if that is indeed the case, then we can diffuse those labels throughout the graph, and that you leverage that information for making new prediction. The second source of prediction power is structural equivalence, which is that nodes with similar label and similar properties in the graph have similar network motifs, similar network shapes occur in their local neighborhoods. And so the idea now is, and a key insight is that when label data are scarce, we really cannot rely on propagating labels because we don't have many labels. So label propagation, it's not meaningful. It's highly unreliable and uncertain. Instead, what we can do, we can leverage structural equivalence and rely more heavily on structural equivalence at that point in time. And in such settings, and local subgraphs can allow us to do exactly that, because they allow us to explore the structure, the network shape that exists in the enclosing subgraph of a target node. Of course, we wanted to go one step further and understand this from theoretical standpoint, and we were able to show that also theoretically, that the influence of a particular node in the target node in the graph, where target node is the node that we want to make some prediction for, the influence of any other node on that target node decays exponentially when that other, depending on the distance between other node and the target node, right? So as we go further away from the target, the influence of those nodes that are further away from the target decays exponentially. And because of that, it makes sense and justified that we can focus mostly on exploring local subgraphs around target nodes, as they contain older or nearly all relevant information. So we were able to show that mathematically, also empirically, the rich body of literature in network biology and medicine has shown in the last years how local information or first order two-hop, second order neighbors are really most useful for making predictions in networks. And so the key takeaway message of of GMeta is that when labels are scarce and label propagation does not work, we can leverage correlation between subgraph structure to make better predictions. And so that has motivated us, and one of that is the core principle of GMeta algorithm that can extract structural information in subgraphs and then transfer that from one label to the other. And that is what we also then used in the COVID study that I have described in the beginning to motivate this work. So let me now speak a bit about the results of that work. So the first question we asked was, well, great, how can we even represent COVID-19? And so back in March, the goal was, or the idea was, to map SARS-CoV-2 targets to human interactome. So to take the viral proteins, identify what are the host proteins, and then map those host proteins to human interactome to identify something that we call COVID-19 disease module. So here in the slide, I'm showing the subgraph of human protein interaction network that is identified by the human proteins targeted by viral proteins. And this key data came from a UCSF lab and was generated by Nevan Krogan's lab and is described in the Nature paper published by Gordon et al., a postdoc in Nevan's lab, I think in April or May of this year. So when we had this, we had this COVID module, at that point that meant we have a great representation of a COVID that is computable for us. We can represent it in the form of a data point that we can then reason about. And so that was great. However, we didn't have any labels for COVID-19. And that's really where GMeta was extremely useful. And we used GMeta in the following way. So the first test, and that all happened between March 23rd and April 15, was let's deploy our algorithms. And we wanted to understand what is possible promise of those algorithms and how potentially they could be useful for. And there was no ground truth information. So the proxy for us was, let's monitor drugs under clinical trials. Why I want to emphasize here, this is really a weak proxy, because drugs that are under test for clinical trials, they only capture the assessment of medical community of what drugs potentially might work rather than what drugs actually work. And that was further even more complicated in the context of COVID because of various external factors. But what the idea was, we took the COVID disease module, protein interaction networks, used GMeta to identify priority list of drugs for COVID, and then evaluate those against drugs under clinical trial at that point in time. So we employed a number of methods. Those here in the green, A1 to A4 are 4 pipelines that are graph neural network pipelines. And then we have many network medicine, proximity, and diffusion methods. This is the A-ROC plot. So it indicates true positive rate against false positive rate. The model that better performing model would be positioned closer to the top left corner. We can see that graph neural network models really performed very well, which was encouraging for us. And so that encouraged then us to share those priority ranked lists of drugs with the scientists at National Emerging Infectious Disease Laboratories, NEDL, which is a level 4 laboratory that can work with live virus. So they were able to test 918 compounds. They screened them for efficacy against SARS-CoV-2, first in African monkey cells, VIRO-E6 cells, and then later on in human cells. The result of that experiment was that 77 drugs have shown strong or weak effects in inhibiting viral growth. They were active across a broad range of concentrations. And from the computational standpoint, that meant that we had an order of magnitude higher hit rate among top 100 drugs their prior work. But so random brute force search would expect to yield around 2% of drugs among top 100 in the predicted list. This ranked list of drugs generated by our AI methodology yield 9% hit rate, which is an order of magnitude better than what would be possible before. And so what we found is that those drugs, those 77 drugs that showed effect on the virus and inhibited the growth in first monkey in human cells, vast majority of those are drugs that exert their effect through networks. So they do not directly bind to proteins targeted by SARS-CoV-2. Instead, they rely on these network-based mechanisms that cannot be readily identified by more classical docking-based strategies for drug repurposing. So that is at a very high level really motivates the use of networks for this problem. Lots of machine learning lessons from this exercise and between March and June. And one really important that I want to emphasize is that when we want to use and implement the AI algorithms in the context of a really experimental pipeline and be able to rapidly and quickly interact with domain scientists, we need to have this, we need to have AI systems that can not only provide predictions, but also explanations for those models, and then being able to integrate feedback provided by domain experts back into the model and improve the model. So we need to have this entire feedback loop, and that is really important. And some of the algorithms that we developed earlier on explainability allowed us to being able to very quickly iterate with domain experts on a daily basis. We literally had daily meetings discussing new results, data sets, predictions, explanations, and so on. Another ML lesson is that the zero-shot learning, which is a top term used to refer to the setting where we need to generalize, where our ML models need to generalize to new diseases. It's really hard setting, in many ways not solved yet. Okay, so I will just conclude here with one particular slide, which is the therapeutics data commons. So TDC, or therapeutics data commons, is a new initiative that we have recently started, and it's a community-driven open source initiative with the goal of bringing the area of therapeutics closer to computational scientists, machine learning scientists who are developing exciting algorithms, so that in TDC, one can find ready-to-use data sets across a wide range of drug discovery tasks, from efficacy, safety prediction, drug response modeling, drug target discovery, and across a wide range of biological products, all the way from antibodies to vaccines and immunos. So we also have a package that is easy to use in 3 lines of code. People can get the data sets and start working on it. And so you're very much invited to check out therapeutics data commons and get involved in some of the exciting ML research, where I think there are many opportunities and many tasks are still underexplored by AI and ML community. I will stop here. All the papers, code, data that I've been referring to and mentioned is on our lab website. Feel free also to send me emails with specific inquiries, and I hope we have a bit of time to answer some questions. Thank you.
Thank you, Marinka. And I think it's a question time. You can just jump in and ask questions. Any questions?
I have a question.
Okay.
I was wondering, in the protein-protein interaction network, if the type of interactions are different, like are they like inhibition, activation, or any type of other interaction? Do you consider them in the message passing?
Yes, so thank you, Negin, for this question. And the short answer is yes. So I didn't show that in the schematic on the slides just because the figures would be very complicated. But what was shown as those black edges between white circles representing proteins, those edges are actually typed. So we have, for every edge, we have an indication that tells us what is the type of that relationship, whether that's a biophysical contact, or whether that is some tissue-specific tissue-specific interaction, whether the interaction actually represents inhibition or activation. And then that is taken into account explicitly during message passing, so that the model, what it does, it asks, oh, given a particular node, what are the neighbors of that node according to a specific type of interactions, right? So what are the neighbors of that node that interact with the node in question through biophysical interaction versus to biophysical interaction, but in specific tissue or specific biological context. And that's really important to consider. Also extremely important was in the context of repurposing work, because domain scientists want to know much more than just saying, here's an association that we predict. A simple pointwise estimate of prediction will not really lead to downstream experimental validation, because it's just high stakes precision without good evidence.
I see. Do you suggest any source to read more about it, its details?
Yes, so that's a great question. So here are some of my suggestions. I'm trying with my lab and my students also to prepare and share lots of these kind of resources, code base and data with the community. So if you go to my lab website, you will find Graph ML tutorials. That's a GitHub repository that has a bunch of end-to-end examples of using graph neural networks on biological networks. So you will see how you can tackle a variety of tasks, and you really have end-to-end examples, just code and data you can readily run. So that's the code base. The data resources, I will check, you go to our lab website and check DDC, Therapeutics Data Commons, which again comes with a package where you can get many of these data pre-processed, cleaned, such that they are done correctly in a meaningful way, and you can get those data sets using 3 lines of code via package that you can install in very simply in Python using pip.
Thank you.
Thank you, Marinka. I have one very quick question. So, well, it was a beautiful talk. And then, of course, applications are absolutely exciting and interesting. So one question that comes into mind is our current knowledge of PPI enough? And then let's put it this way, that if we were going to use, let's say, half of the knowledge that we have to use the graphical neural network that you have, would we yield the same accuracy of the prediction, and how reliable that would be?
Great question. I have 2 answers to that. So first answer is, I would say that solely considering human protein interaction network, the way it's given to us at this moment or these times is not sufficient, right? So the human reference protein interactome, one that was published in April by Mark Vidal and other in Nature, and many other efforts essentially show that we are on the range of 20–30% of proteins being tested for interaction. So because of that, the network is severely incomplete. And we are trying to and are working a lot with knowledge graphs now, or heterogeneous networks, where we have these multi-scale models. So one project, I didn't go to it right now, but it's an ongoing work where we have protein interaction network and then also single cell data sets to try to map these interactions and enrich them to with information about cell types and tissues and then other signaling types of networks. So I think that for many problems, protein interaction network alone is not sufficient, because it is not the only layer of information, of course, that would explain biological complexity, which was part of the motivation of the talk, plus the network is fairly incomplete. The second part, which is we did a lot of experiments. So every time we do a particular study, we do these kinds of ablation studies where we ask ourselves, how does performance of the model vary when we hide part of the data? So essentially generating those curves when on the x-axis we say, what happens if I have only 10% of the data, 20%, 30%, 40%, and so on? How does the performance change? And we do that not only by randomly hiding part of the edges, but then we typically also do that in some meaningful way. So when we were testing GMeta, we designed train splits when we removed from the PPI network all edges done by or associated with a particular set of cell lines, and then see, well, how does that affect the model? So not just randomly removing edges and examining how that affects the model, but also doing that in a more biologically meaningful way. And the key message there is that many predictions made by the models today, and like if we go and read papers, we have these papers were reporting astounding accuracies, 90–80% of AUC scores. However, when we really use those models in practice, we get near random predictions. And so when we were looking quite a bit into that in the context of therapeutics problems, and it turned out that these models work really well and make technically correct predictions, but they are trivial. So what that means is that they're trivial in the sense that while that might be a molecule, but it turns out that it's an explosive. Or like, while that might be a molecule, but it's trivial in the sense that it's a very slight variation of what is already in the training data. So that indicates that what the model has done, it has memorized more or less what it has seen, and it will propose some variations of the data that it has seen, but not really generalize to new problems. And I think that explains that's one of the reasons why we see this gap between computational ML papers reporting astounding accuracy metrics, and then what happens when we really use these methods in real work, right? When we really put them into work with our collaborators and domain experts, and then you see, well, that there is this huge gap that exists there.
Right. Yeah, and yeah. So this is a great talk. I'm asking a question for Professor Pan Li, who also from Yuri's lab. You must know him. And he has another meeting to run actually. So I asked question for him. He asked like I think the question is about it's a little bit technical. It's about your anchor patch. So how large is the inference for this anchor patch selection for your final performance? For example, I can imagine that you have you can select some random nodes and select some random subgraphs as like your anchor patch, but like they may have some overlap. Or how do you select that? Do you need to first run some network community detection, and on each community you're going to select some of the anchors?
That's a great question. So we avoided the use of network community detection algorithms because using those algorithms in the anchor patch sampling strategy would prevent us from being able to propagate gradients back to the model. So the model would simply not be differentiable. So instead, what we have three distinct anchor sampling functions, Y3, one per each of the three aspects of subgraph topology: neighborhood, structure, and position. And so those functions determine exactly how anchor patches are sampled. The fact that they were anchor patches that are sampled might be overlapping is not an issue. That's actually a desired or a positive effect, because in that case we also simulate real-world subgraphs which overlap, because we know that some drugs, their targets, some diseases have common genes. So we have these overlapping subgraphs. So it's actually good that anchor patches are overlapping as well.
I see. Yeah. I got it. I can explain for you. Okay. Is there any more questions? No? Yeah, I think we are right on time, actually, a little bit over. If there's no more questions, I think that's today's talk. Let's thanks again for Professor Marinka Zitnik for his wonderful talk. It's really, really insightful. I already have a lot of ideas, given like your like beautiful projects about like a drug side effect, adversarial effect, and also the subnetwork embedding. And if you want to work with her or have any questions, you can directly email her, I guess. Okay.
Thank you.
Okay. Today's talk. Thank you so much for to give us this talk. Thank you.
Of course. Thank you for inviting me, and I look forward to future interactions. Thank you.
Yeah, exactly.